body. However, there are several differences in how the two systems exert their control. The chemical messenger of the endocrine system is a hormone, which is released by glandular cells and diffuses into blood vessels serving the endocrine gland. Once in the bloodstream, hormones are transported throughout the body and have a relatively widespread effect on distant cells. Compared to the nervous system, the stimulus response time for endocrine control functions is slow and prolonged. It may take hours, even days, for the effect of a hormone to be noticed, and the effect may persist long after the hormone has been removed. In contrast, communication in the nervous system is by way of electrochemical impulses conducted by neurons that cause the release of chemical messengers called neurotransmitters from the axon terminals of the neuron. The neurotransmitter is released into a space called the synapse and diffuses across the synapse to a postsynaptic cell on which it exerts a local effect. The stimulus response time for nervous control is very rapid, on the order of milliseconds and of short duration. The first experiment in this series is designed to demonstrate the effect of pituitary hormones on egg production in the frog. Despite its small size, the pituitary gland produces a wide variety of important hormones. Two of the hormones produced by the anterior pituitary gland regulate the ovarian cycle in the female. These hormones are follicle-stimulating hormone, also called FSH, and luteinizing hormone, referred to as LH. The hormone FSH primarily affects follicle development in the ovary, and the hormone LH stimulates final maturation of the follicles and release of eggs from the follicles in a process called ovulation. In this demonstration, two gravid female northern leopard frogs are used. Before treatment, both frogs are examined for evidence of ovulation. This procedure involves holding the frog firmly with one hand and gently squeezing the abdomen toward the direction of the legs. If eggs are fully developed and ovulation has occurred, pressure on the abdomen will force the eggs out of the oviduct through the cloacal opening. Since neither frog has been treated, no eggs are expressed through the cloaca in either animal at this time. One of the frogs, the experimental animal, is injected with commercially prepared frog pituitary extract into the lower quarter of the abdominal cavity. The other frog, the control animal, is injected with the same volume of normal saline solution in a similar manner. Following injection of the animals, they're returned to separate jars containing a small amount of pond water. The animals are allowed to remain in the jars undisturbed for about 24 to 48 hours. After the elapsed time, each frog is again checked for the presence of eggs in the cloaca. When the abdomen of the control frog injected with normal saline is squeezed, the results are again negative. However, when the abdomen of the experimental frog injected with pituitary extract is squeezed, eggs are expressed through the cloaca in a continuous stream of gelatinous material. The hormone in the pituitary extract which induced ovulation was luteinizing hormone. In the next experiment, the effect of hyperinsulinism will be demonstrated. Insulin is produced in the pancreas by glandular cells located within the pancreatic islets. Insulin increases glucose uptake by most cells of the body. However, insulin is not required for glucose uptake by nerve cells in the brain. If excess insulin is present, a condition called hyperinsulinism, glucose uptake by muscle and most other cells is increased, resulting in a dramatic fall in circulating glucose levels. If circulating levels of glucose decrease significantly, a condition called hypoglycemia, brain neurons become starved for glucose. In this experiment, the effects of hyperinsulinism on a goldfish will be examined. The bowl marked A contains 400 international units of commercially prepared insulin in 100 milliliters of water. Bowl B contains 100 milliliters of a 10% glucose solution. A small goldfish is placed in bowl A containing insulin for observation of changes in behavior over time. Insulin is taken up rapidly across the gills to create a state of hyperinsulinism. Within 5 minutes, the body of the fish begins to twitch and movements become increasingly convulsive. Rapid darting swimming movements are observed for a few minutes. Eventually, a slower, regular, circular swimming pattern resumes, but the fish is unable to maintain an upright position. For several minutes, the fish frequently turns over on its side while swimming, but continues to attempt to upright itself. Within 20 minutes, the fish remains on its side, stops swimming, and finally enters a comatose state. The change in swimming pattern and the final comatose state are the result of brain deprivation of glucose produced by hyperinsulinism. The comatose fish is then carefully transferred to bowl B containing the 10% glucose solution and again observed for changes. Within a few minutes, the fish begins to revive. And within 15 minutes, it resumes its normal swimming activity. Placing the fish in a 10% glucose solution restored circulating glucose to a level necessary for normal brain activity. The results of this experiment should clearly illustrate why human diabetics who rely on insulin therapy must carefully regulate the amount of insulin taken and be attentive to early signs of hypoglycemia. In the final experiment, the effects of epinephrine and acetylcholine on the heart will be demonstrated. Epinephrine and acetylcholine are chemical messengers of the autonomic nervous system. The primary source of circulating epinephrine is the adrenal medulla, which is an extension of the sympathetic division of the autonomic nervous system. Epinephrine released from the adrenal medulla has a stimulatory effect on both heart rate and force of contraction. The sympathetic division also exerts a stimulatory effect on the heart through cardiac sympathetic nerves that directly innervate the heart and release the chemical norepinephrine, a neurotransmitter which is chemically related to epinephrine. Acetylcholine is a neurotransmitter secreted by the parasympathetic nerves that innervate the heart. The major effect of acetylcholine on the heart is to decrease heart rate by affecting the pacemaker cells of the heart. The central nervous system of this bullfrog has been destroyed by decapitation and pithing, and its ventral body cavity exposed. The three-chambered heart of the frog can be seen beating within the glistening pericardial sac.
The brain's gone. The heart keeps going.
In this view, the pericardium has been opened and the heart has been linked to a pen rider for recording force of contraction and heart rate. If the heart is kept moist with Ringer solution, the heart continues to beat, and the number of beats counted or recorded on a pen rider in 1 minute represents the baseline heart rate. For this demonstration, the pen rider chart speed has been set to 2.5 millimeters per second, and the interval shown on the record represents a 30-second period. Each large vertical deflection represents 1 heartbeat. Since there are 19 heartbeats during this 30-second time interval, the baseline heart rate for this frog is 38 beats per minute. After allowing the heart to beat at baseline rate for several minutes, the heart is flushed with a few drops of a 1 to 1,000 solution of epinephrine. less than 10 seconds, a change in the heart rate and force is observed. In this trial, heart rate increased from 38 beats per minute to 48 beats per minute. The force exerted by the heart more than doubled under the influence of epinephrine. This is recorded on the penwriter tracing as an increase in the height of each wave. The increased strength of contraction is also evident in the size change of the heart and changing color of the heart muscle as blood is squeezed out with greater force. For the second half of this experiment, a frog heart has been linked to a penwriter in a way that records the atrial and ventricular contractions of each heartbeat. Although it may appear from the record that the baseline heart rate of this frog is twice as fast as that of the previous frog, the baseline heart rate is actually only 46 beats per minute, since each pair of deflections represents a single heartbeat. Once a new baseline heart rate has been obtained, a few drops of a 1 to 50 solution of acetylcholine are placed on the heart. Heart rate begins to decrease dramatically within 10 seconds. In this trial, heart rate decreased from 46 beats per minute to 12 beats per minute in the first minute following administration of acetylcholine. The decreased heights of the atrial and ventricular waves indicate that force of contraction also decreased as a result of acetylcholine administration. The results of this pair of experiments illustrate the opposing and differential effects of epinephrine and acetylcholine. Epinephrine increases both heart rate and force of contraction, but has a more pronounced effect on force. Acetylcholine decreases both heart rate and force of contraction, but has a more pronounced effect on heart rate. In multicellular organisms, control and integration of cellular function by chemical messengers is essential to survival. In this series of experiments, some of the effects of selected chemical messengers of the endocrine and nervous systems have been demonstrated. Control of ovulation by LH, regulation of glucose levels by insulin, and control of heart function by epinephrine and acetylcholine. These experiments illustrate only a few of the many important functions regulated by chemical messengers of the endocrine and nervous systems, the major controlling systems of the body.